Good day, Philippines! This is Biotech Report for today. In the Philippines, banana is currently grown with total area of about 35,000 hectares, which is estimated 3% of total arable land. For the past years, the area planted to banana has increased, which is indicative of growing economic importance of horticultural crops. For several years now, banana growers in the Philippines had been quietly battling a plague. A Panama disease is a fungus formerly known as tropical rice 4. It has killed more than 15,000 hectares worth of Cavendish bananas in Davao region, where most of areas are cultivated. Lakatan, which is the most common cultivars of banana in the Philippines, is also susceptible to Panama disease, or also known as banana wilt. It is a devastating disease caused by soil-inhibiting fungus species Fusarium oxysporum. Using trichoderma and synthetic fungicides are some existing solutions used by some lakatan growers. Trichoderma is a potential and effective source against notable pathogens. For many years, synthetic fungicides had been used to control plant diseases. Mancozeb, Tyram, and Detain are some examples of fungicides readily available in the market.
Although synthetic fungicides are highly effective, their repeated use has led to problems such as environmental pollution, development of resistance, and residual toxicity. In addition, due to this issue, we proposed a new solution for our lakatan banana growers. This technology is called Trangenic lakatan. Genetic engineering of banana is considered a perfect alternative for improvement of sterile cultivars or ones which are not amenable to traditional breeding methods. The main steps involved in the development of these GM crops are isolation of the genes of interest. It is existing knowledge about the structure, function, or location on chromosomes is used to identify the genes that is responsible for the desired trait in an organism. For example, drought tolerance or insect resistance. Next is the insertion of the genes into a transfer vector and plant transformation. It is the most commonly used gene transfer tool for plants. It is a circular molecule of DNA or plasmid from the naturally occurring soil bacterium, Agrobacterium tumefaciens. The genes of interest is inserted into the plasmid using recombinant DNA or rDNA techniques. Next is the selection and regeneration of the modified plant cells into whole plants. For this method, genes responsible for resistance are inserted into the vector and transferred along with the genes conferring desired traits to the plant cells. When the cells are exposed to the antibiotic or herbicide, only the transformed cells containing and expressing the selectable marker gene will survive. The transformed cells are then regenerated into whole plants using tissue culture methods. Information about the marker genes and whether they will be present or absent in the developed GM plant is provided to the regulators. Next is the verification of transformation and characterization of the inserted DNA fragment. Verification of plant transformation involves demonstrating that the gene has been inserted and is inherited normally. Tests are done to determine the number of copies inserted, whether the copies are intact, and whether the insertion does not interfere with other genes to cause unintended effects. Testing of gene expression is done to make sure that the gene is functional. Testing of plant performance. Testing of GM crops is an important issue for the legitimacy, biosafety, and regulatory purposes. The area under GM crops is increasing very rapidly, and many new genes are being introduced in major crops. Last is the safety assessment. Food and environmental safety assessment are carried out in conjunction with testing of plant performance.
For the safety of humans, environment, animals, and other related microflora, a comprehensive molecular testing of newly developed GMO is very important before commercial release. Regulatory process for GM crops approval need comprehensive risk analysis for each case separately. The detection and identification of GMO is also a great value in identifying the purity of sample, labeling food, and trade reason. Therefore, combined use of more than one testing method would be advantageous for complete analysis, authenticity, and biosafety assessment of GM samples. The regulatory process starts of preparing a project proposal for submission to the Institutional Biosafety Committee. After submission, a proposal to IBC, which conduct a risk assessment and endorse to NBCP, we can now apply for a permit to conduct contained testing. After contained testing is complete successful, we can apply now to DA BPI for a field testing permit. The DA BPI creates a STRP current with public notification, and IBC and STRP evaluates potential adverse effect to the humans and environment is called risk assessment by STRP, and the BPI Cure Biotechnology team after receive a field test permit and each field is evaluated. We can now obtain permit for release, which is the propagation and commercialization. That's all. Thank you.